2002, Dr. Walters joined the University of Minnesota as an assistant professor and was promoted to associate professor with tenure in 2008. During this time, her lab studied the mechanism by which ubiquitin signal is for degradation by the proteasome. Dr. Walters was also awarded an American Cancer Society Research Scholarship from 2007 till 2011 for her work in protein quality control. In 2013, she moved to the Center for Cancer Research as a senior investigator, where she runs a vibrant research program that aims to apply basic science discoveries to the translational benefit in the clinic. She currently serves as the CCR Science Board and as an acting chief of the Structural Biophysics Laboratory. And the title of her talk today is Outskirts of the Proteasome, Substrate, Receptor, and Beyond. I invite Professor Kylie Walters on the stage.
Thank you very much for that kind introduction, and I'm honored to speak here today. Thank you very much for the organizers for this opportunity to share our research. Hold on. Okay, I guess I clicked to get it to forward. So the work that I'll be showing today was done by these stellar scientists, and I'll kind of try to acknowledge them as I go along, but I'll have their names at the lower corner of my slide so you can see who did the work. But Vastiosai Amponsa, Shushu Liu, Shang Chen, and Gwen Buell all contributed to the work that I'm going to show today. And so a couple weeks ago, Ranavir gave a wonderful talk talking about the ubiquitination pathway, talking about the enzymes involved in ubiquitinating substrates. So I won't belabor this pathway, but just to remind you that there's an enzymatic cascade that leads to substrate ubiquitination. And then in some cases, that becomes a signal for targeting to the proteasome. And then there are substrate receptors that recognize the ubiquitin chains in the proteasome. And then the ubiquitin chains on the substrates get trimmed back by deubiquitinating enzymes. And at the same time, an ATPase ring engages the substrate and starts to feed it into the core particle of the proteasome where the catalytic proteolysis occurs, and then the substrate gets proteolyzed into smaller peptides. And so for this talk, I'm really going to be focusing on this stage of the ubiquitin proteasome pathway, how the proteasome comes to recognize its ubiquitinated substrates. And so I'll just briefly go through the three major receptors for these substrates that are in the proteasome. And so one of them is RPN1. It's the most recent one that we, in collaboration with Dan Finley at Harvard, was able to establish as a substrate receptor. It has two regions that participate in this activity. One is we named a T1 site, T for toroid, because it's in the toroid structure of RPN1. And you can see it kind of indicated here in blue. And then on this kind of showing just the top capping regulatory particle on the proteasome, you can see where RPN1 maps by cryo-electron microscopy reconstructions. And then we were able to solve the NMR structure of this region of RPN1 bound to K48 linked ubiquitin chains. And you can see here, when I go forward and we're kind of talking about the ubiquitin chains, there's what we call a proximal end. That's the end that's closest to the substrate that would have a free glycine 76 that could attach to an amino acid on the substrate, and then a more distal end, which is further away from the substrate. And so what you can see here is that there's two regions in the RPN1 T1 site that can engage the ubiquitin moieties, and they can come in either orientation with the proximal one here or the proximal one here. And then also in RPN1 is another region that recognizes the ubiquitin fold, but it recognizes the ubiquitin fold in one of the 3 deubiquitinating enzymes, USP14. So at the same time that there's a site to recognize ubiquitin and ubiquitinated substrates, there's another site that recognizes the deubiquitinating enzymes, which of course have to start to trim back the chain. And so the first actually discovered substrate receptor in the proteasome was RPN10, and so I'm showing it here. It has this well-ordered VWA domain that you can see in this cryo-EM reconstruction here, where it would locate. But then following it are flexible linker regions with a lot of intrinsic disorder. But there's these, in the human RPN10 protein, 3 helices and 2 ubiquitin interacting motifs, or UIMs, and you can see them bound to, each of them binds to a ubiquitin moiety of a ubiquitin chain. You can see it bound to K48 linked diubiquitin chain, and we are able to also solve this structure by using NMR. So NMR has just been a really good tool for looking at these complexes with ubiquitin. And because they're so dynamic, and because, and this one, like RPN1, can also bind in the other orientation, it's very hard. They can't crystallize, and they're too small for cryo-EM. So in fact, in this RPN10 structure here, you're seeing the VWA domain, but this disordered region has not been seen to date by cryo-EM because it's got too much conformational heterogeneity. And then the third receptor that we are able to establish as a ubiquitin receptor in the proteasome with Yvonne Dietrich's lab and also Dan Finley's lab is RPN13, and you can see the structure here of the region involved in that. So this is, we call it a Pru domain, and it's seated up here in this region of the regulatory particle of the proteasome. And what you're seeing here is its structure solved with ubiquitin. And then coming in orange, coming off of this large protein, it's like 100 kilodalton protein that has an intrinsically disordered region at its C-terminal end, and it's that intrinsically disordered region that comes and kind of snakes along the RPN13 structure, as you see here, with quite high affinity, 14 nanomolar affinity. And so RPN13, its full structure, this is the region I just showed you that combined to ubiquitin up here and the proteasome down at this end of this domain. It also has another domain that's been named the DUBAD domain that contributes another deubiquitinating enzyme to the proteasome. So if you recall in the previous slide, I talked about how RPN1 contributes USP14, a deubiquitinating enzyme, to the proteasome. RPN13 contributes UCH37, also called UCHL5, to the proteasome by binding to this domain. And by itself, these 2 domains can interact with each other. And so you're seeing here the structure of RPN13 by, just by itself, not bound to anything else. And so with 3 major receptors for ubiquitin chains in the proteasome, of course, a fair question is how important really is RPN13? And Vasti was able to get at this in part by using CRISPR-based cell lines. So one of them is simply missing RPN13, so delta RPN13 knocked out of the cells. And then the other one is missing one of the exons that are in this Pru domain. And so it ends up making a truncated protein. And this truncated protein has a defective Pru domain, and it can't bind the proteasome. We can see that it can't bind the proteasome in cells because if we immunoprecipitate the proteasome using an antibody against one of the ATPase subunits, then we can see the wild type RPN13 protein there, but not this truncated RPN13 there. And So then if we go and look and say, well, now how important is RPN13? Does it affect ubiquitin chains being at the proteasome? Then you can indeed see an effect. So this is the wild type protein looking at how much ubiquitin chains are at the proteasome. And this is what occurs when you lose RPN13 completely. And this is what occurs when you have the truncated RPN13. And this is not because of losing the deubiquitinating enzyme that it contributes, because if you just lose the deubiquitinating enzyme, you actually get an accumulation of ubiquitin. So kind of this cell biology experiment suggests RPN13 is playing an important role at the proteasome. And of course, what we were interested in is what could this role be, and to try to get at that explicitly by using NMR to solve the structure of RPN13 complex with these ubiquitin chains. And so Shushu Liu picked this up. And so what you're seeing here is a half-filtered carbon-13 nosy experiment with RPN13 labeled, and then the proximal ubiquitin of the K48 linked diubiquitin chain unlabeled, and the distal one carbon-13 labeled, and then also that that 14 amino acid peptide that's extended off the proteasome that is used to recruit RPN13 to the proteasome is also present. And so in a half-filtered nosy experiment, we can see interactions between RPN13 and the proximal ubiquitin, and of course RPN13 and the peptide. And so if you look at this, you can see lots and lots of interactions. The ones labeled in gray are to the proximal ubiquitin, the ones labeled in orange are to the peptide that it binds. And then we can do the exact parallel experiment, but in this case, we have the proximal subunit of ubiquitin carbon-13 labeled and the distal one unlabeled, because we just synthesize these chains enzymatically so we can exactly control which subunit of ubiquitin is labeled. And when you look at the NOEs that occur, they mimic the proximal one for the distal, but they're weaker in intensity. So if you look at kind of these signals that are coming through, you can see that they're really very similar, but that they're a little bit weaker. And so what this immediately tells you is that RPN13 is able to bind to either the proximal or the distal ubiquitin in the K48 link chains. And it does so using kind of a common binding mechanism. And this is consistent with what we saw with RPN1 and RPN10, where we could again see a very dynamic interaction where the ubiquitin could bind in either orientation to either of two sites. In this case, however, RPN13 has just one site, and so it would appear that it's exchanging between these two locations at the proximal or the distal subunit of a chain. And so Shushu went forward and calculated what does the structure look like when you input all the NOEs that we were able to get by using the carbon-13 half-filtered nosy experiments. We also use, of course, just N15 dispersed nosy experiment, carbon-13 dispersed nosy experiment, and got a wealth of distance constraints, and then able to get the structure of each complex. And so what you're seeing here is, in this case, the RPN13 is bound onto the proximal ubiquitin. And in this case, the RPN13 is bound onto the distal ubiquitin. In orange is that 14 amino acid peptide that it uses to bind to the proteasome. And then coming off is a distribution of a conformational ensemble that the unbound ubiquitin in either case can adopt. So there is some order around the linker region, and I'll show that soon, but for the most part, there's a widespread in conformation that the unbound ubiquitin can indeed adopt. If you overlay these two structures, then you see, as expected from the NOE interactions that I showed you, that it overlays quite well, and that when RPN13 is bound to either the proximal or the distal, it's quite similar in the interactions. And we can kind of zoom in here and just zoom in on RPN13 with the proximal ubiquitin, and it's the same for the distal ubiquitin, where leucine 8, valine 70, isoleucine 44, which are well known in ubiquitin to bind binding partners, receptors, are playing a role in RPN13 binding. And also this phenylalanine 76, we know from an earlier study that we did that if you mutate that phenylalanine to an arginine, then RPN13 can't bind to ubiquitin anymore. So everything kind of matches up as expected. But what's really interesting is how RPN13 dynamically interacts with each of these ubiquitin moieties in the K48 link diubiquitin chain. And so one really interesting phenomenon that we were able to observe in our spectra was because we have a bound, so for the proximal or the distal, we have a we have a signal for both the bound state and also the free state, because when RPN13 is bound to the proximal ubiquitin, the distal, for all intensive purposes, is in a free state. Also, there could be exchange coming on and off where there is some free state ubiquitin. And indeed, we collected data with the K48 linked diubiquitin at 1.2-fold molar excess. And even in cases where it was in excess, we were able to, well, I'll kind of get to that a bit later, but we were able to see kind of this dynamic exchange going on. And so you can see it here. If we take, for example, this alanine 46, beta and alpha state, we can see signal for both the bound state and the free state. And this is with the proximal ubiquitin labeled, this is with the distal ubiquitin labeled, and we can see interactions going on between these states. So these kind of exchange NOEs in our carbon-13 edited nosy experiments. So within the time course of this experiment with 100 millisecond mixing time, we can see this exchange occurring. And so one of the features of RPN13 in an earlier study, Sheng Chen was able to look at RPN13 binding to ubiquitin of all the different possible linkages of the chain. So the way ubiquitin forms a chain is the C-terminal glycine forms an isopeptide bond to any of the lysines or the N-terminal end of another ubiquitin. And so you can see all of those chain possibilities in this assay. And what's kind of apparent here is that RPN13 binds most strongly to K48 linked ubiquitin chains. And so when we look at our structure, we can understand why. So in this linker region, so what you're seeing here is glycine 76, which is linked to, it's the distal ubiquitin glycine 76, which is linked to lysine 48 of the proximal ubiquitin. And what we can see there is a hydrogen bond forming between that glycine 76 and in RPN13 lysine 103. So and also we can see other interactions going on in the linker region, but this is kind of a really prominent one. And we can see, you know, evidence of that from all of our different NOEs, kind of looking at lysine 48, glycine 47, and kind of looking at all the interactions that are occurring in the linker region that would not be able to occur with monoubiquitin, or if the ubiquitin chain were linked through a different lysine. So this structure kind of speaks to why there's a preference for the lysine 48 link chain. But there's also additional information we were able to get out unique to the lysine 48 link chain. So although you saw that kind of wide distribution for the unbound ubiquitin locally in the linker region, there is some order, and that comes from this leucine 73 of the distal ubiquitin comes up and interacts favorably with, for example, the tyrosine 59 of the proximal ubiquitin. And you can see this through the NOE interactions. You can see leucine 73 interacting with lysine 48 of the proximal ubiquitin, and then surrounding amino acids. So there's also unique to the K48 diubiquitin kind of local structure that it forms around the linker region that only lysine 48 link chains could do. So we were, of course, curious, does K48 link diubiquitin form this configuration in the absence of RPN13? And crystal, lysine 48 link ubiquitin has been studied for a very long time, and crystal structures were solved a long time ago of it. And so a very well-known crystal structure of lysine 48 link diubiquitin is this one shown here, where you can see kind of this packing of the ubiquitins against each other. In this configuration, RPN13 would not be able to bind to the chain because its binding surface, which as you recall had leucine 8, valine 70 in it, they're not exposed because the ubiquitins are packing against each other. Also, there's a crystal structure where there's also packing a bit of the two ubiquitins against each other. And this was named open, although you can see there's packing of the ubiquitins in this crystal structure too. So what Shushu did was to look at just free K48 link diubiquitin. And in this case, what you're seeing here is one of the spectra whereby the proximal subunit is carbon-13 labeled and the distal one is not labeled. And then again, taking a carbon-13 half-filtered nosy experiment and looking what NOEs you see. And what's apparent is the NOEs consistent with this structure are present in the NMR spectra. So certainly this closed conformation for K48 linked diubiquitin, we can see depicted in our NMR spectra, and where it's labeled kind of the brown against the blue, then that's consistent. But what we can also see are NOEs like what I showed you previously that we observed with RPN13 present. So between leucine 73 and the linker region. They're smaller, so it's a more minor population, a more minor state of K48 linked diubiquitin compared to this state, but it's present. We don't see any evidence of this state in our K48 linked diubiquitin spectra. And so what this data suggests is that there's a dominant form in solution for K48 linked diubiquitin with the two ubiquitins packed up against each other, but then also this form where there's interactions around the linker region and there's an ensemble of configurations in terms of how the two ubiquitins distribute relative to each other. And it's this form that RPN13, of course, is able to select. And so this is kind of the model we have, putting together all of this data so that the K48 linked diubiquitin in solution exists in at least these two states. What we can observe are these two states. We can observe a dominant form where they're packed against each other. The two subunits are packed against each other. A more minor state where there's interactions around the linker region and there's a distribution of the ubiquitin configurations relative to each other. And when you have RPN13 present, it selects this state. It can't bind to this state, and it's selecting this state and driving it into this configuration. And meanwhile, RPN13 can exchange between the subunits. It prefers to be on the proximal one because of those interactions that it has with the linker region, but it's capable of exchanging. And and as I kind of alluded to before, when we had the K48 linked diubiquitin in excess, and we record spectra looking for interactions between the two ubiquitins, we never see it go back to this state, at least in the time course of our experiment. So we see that when RPN13 is present, it keeps the K48 link chain in this kind of opened configuration through its dynamic interactions. So what could this mean physiologically? Well, by RPN13 keeping it in the chain in this more open configuration, it's going to be easier for the other receptors to interact with it, such as RPN1, that's also in the proteasome. It's going to also have the chain more opened, which could help with the activity of deubiquitinating enzymes is particularly it contributes UCH37, UCHL5 by its C-terminal do-bad domain. And so maybe keeping the chains more open helps with hydrolyzing the chains because the substrates can't translocate into the proteasome core particle with all the ubiquitin chains on them because it can't fit through the narrow gate. And then shifting, so then this I'll talk a little more about in the next slide, but I think what's really compelling is that by having the ubiquitin chain in this very dynamic state, it might also help promote movement of the substrate. And so we just had this work published this year in Structure, and we were very lucky that Jeroen Roloff wrote a really nice kind of review or preview for our paper, and he put together this really nice image. I'll show you ours later. I think his is easier, so I'll start with his. So he kind of shows kind of the model that we're getting at where RPN13 is interacting with the ubiquitin chain and keeping it open. And in these dynamic interactions that also occur at RPN10, oops, sorry, I don't know how to go back though, RPN10 and RPN1, then the substrate also is moving around. This movement of the substrate is important because the substrate has to come in and engage the ATPase ring. And so here's kind of a little bit more complicated model that we put together is that we believe that the substrates would have multiple ubiquitin chains on them in many cases, and that these dynamic interactions similarly, like kind of maybe more easily visualized here, allow the substrate to move around until the substrate's able to engage productively with the ATPase ring. And then, but in this way, kind of the model is more like kind of sliding and hopping around the chain like many other biological systems in nature, such as, for example, DNA repair enzymes, where they're kind of moving around until they hit a site that needs repair. And so we think that the dynamics is really critical to what goes on in the proteasome. And so now I just want to switch to RPN10. RPN10, I already talked a little bit about that it has a domain that binds to the proteasome and then two UIMs that bind to ubiquitin. And what we noticed kind of when we went and lined up in higher eukaryotes that there's another C-terminal region that also was conserved. And this was picked up by Gwen Buell and Shang Chen. And we went through, I won't show you all the data, but we found out that this C-terminal conserved region doesn't interact with the UIMs. It doesn't bind ubiquitin. And when we then went and did mass spectrometry and pulled out from whole cell lysates what it interacts with, the mass spectrometry pulled out E6AP, also called UBE3A. So it's an E3 ligase that's associated with Angelman syndrome, cervical cancer, and autism. So we were really excited by this. And so this is E6AP. And going through and figuring out which domain it bound, this is kind of showing you the NMR proton nitrogen HSQC experiment. In black is the free C-terminal region of RPN10 that I showed you had conservation, and we picked out it binding to E6AP. And then you can see in green is when we add in twofold molar excess of this N-terminal azul domain. And you can see really dramatic shifting of peaks. They become very much more dispersed. So this is, and I'll get to that a little later, this is the azul domain. The structure was actually solved in Cheryl Arrowsmith's lab as part of the Structural Genomics Consortium, and it was really not known what the function, it hasn't been known what the function of the azul domain is, and so it's really interesting that it binds, that it's binding to RPN10 C-terminal region, and then when we did ITC and surface plasmon resonance, we get a really strong affinity. The affinity is down at about 10 nanomolar. And so then I think I'm getting close on time, so I won't go through this, I won't belabor this too much, but just to say that when we overexpress E6AP, or RPN10 in cells, we can pull out whichever one's endogenous protein. So if we are overexpressing RPN10, then we can pull out endogenous E6AP. If we're overexpressing E6AP and RPN10, then again, we can see their interaction. So this kind of shows that they're interacting also in cells. And so we named this domain in RPN10 the Razul domain for RPN10 Azul binding domain. And so we wanted to know, of course, what the functional significance of Razul is. One of the features that we noticed is we were able to make a CRISPR cell line where we were missing that C-terminal Razul domain. And when we did so, the RPN10 truncated delta Razul construct is at lower levels, and so correlated the E6AP levels were also lower, and we could confirm this by RNAi. So if you knock down the RPN10, kind of here, this is by RNAi knocking down RPN10, then you can see corresponding low levels for E6AP, but not the other way around. If you knock down E6AP, you don't affect RPN10. So this really even further linked these two proteins. When we go in and try to rescue, we can't just rescue by overexpressing, but what we did find is that when you try to just express E6AP in cells, it expresses at very low levels unless you also co-express RPN10. So the production of these E6AP seems to be dependent on RPN10. So up until now, for the RPN10 part of the story, I've showed you that it has a C-terminal region that we've named Razul that binds to E6AP Azul domain. It binds strongly at 12 nanomolar affinity. When you lose RPN10, you lose correspondingly E6AP. And when you want to express E6AP, you need RPN10 around to drive high levels of expression. So with all of this, a critical question is, does the Razul domain in RPN10 contribute E6AP to the proteasome? And I'll show you that it does. So this is kind of a control, IgG control, looking at how much E6AP is at the proteasome when you don't have RPN10 around. And then this is going through and looking at immunoprecipitating out the proteasome with either the delta Razul expressed or the full-length RPN10 protein expressed. And you can see E6AP is boosted at the proteasome by the full-length RPN10. And when you overexpress the Razul peptide, then you lose E6AP from the proteasome. So the effect is modest, again, because the levels of these proteins are really dependent on each other. So when we're working in this RPN10 cell line where you have the truncated protein and it's at really low levels, then E6AP is also at low levels. But nonetheless, we feel that this is sufficient evidence to say that Razul is playing a role in contributing E6AP to the proteasome. So we were really interested in, of course, getting the structure of E6AP at the proteasome, but this was complicated by, again, the intrinsic disorder and the limitations of cryo-EM for regions with intrinsic disorder and structural heterogeneity. So we tried, what you're seeing here is kind of a mapping of the resolution as you go out to the periphery of the proteasome, the resolution gets really low, and we, like others, couldn't see the RPN10 UIMs. What is kind of, and we couldn't similarly see E6AP, it would be too heterogeneous. What we could see that's going to come out, I think, next month in structure is unanchored ubiquitin chains kind of in this central region of the proteasome binding to a deubiquitinating enzyme in the proteasome. And we think that the RPN10 UIMs might be contributing to the recruitment of the chains at this site, but I don't have time to talk about this here. I'm going to go back more to the data that we do have on E6AP.
Heidi, you have already exceeding your time, so maybe you can wrap up if you don't mind.
Okay. I'll just quickly show you, these are 1D traces where you can see that the E6AP at the proteasome by NMR, and then I'll just go to the structure that we did by NMR, that Shang did. And you can see it's kind of cool. It forms this 4-helix bundle. And what's interesting is you probably picked it out that it becomes more dispersed with the azole domain, and that the binding induces helicity and structure. And we are also able to confirm that by circular dichroism. And so this is just kind of the model that it's E6AP is recruited to the proteasome through this azole domain. And I just once again want to thank Gwen, Sheng, Shushu, and Basti. So sorry I went over my time.
No, that's okay. Thank you so much for this exciting and very stimulating talk on two different stories. I'm sure audience has questions, and we can take a couple of questions maybe before we move to Mark. I don't see a single hand raised so far. Okay, so before people,
Ranveer has raised his hand, Banda.
So, who?
Ranveer. Yeah.
Ranbir, you can go ahead. You can ask a question. I don't see it here though.
Thanks, Mandar. Kylie, that was fantastic. I didn't know about the story of the azole domain. I'm wondering, what is the E6AP doing at the proteasome?
Yeah, that's a great question, Ranavir. So the model that we have is that, well, first of all, I should just say we don't completely know, but one compelling model based on all the evidence in the field is that it's reconfiguring the ubiquitin chains, and that it could be playing an important role in making sure that you get a really efficient chain landscape on a substrate. So I'm sure you're familiar with the papers that came out that have shown that kind of these heterogeneous mixed link chains are really good, efficient substrates for the proteasome. And so E6AP could be playing a role that, you know, a substrate comes into the proteasome, it may not be ubiquitinated in the way most efficient for degradation of the substrate. There's also a little bit of evidence it could be playing a role in directly ubiquitinating components of the proteasome. And it may be that that happens under certain cellular conditions. We looked for that. We couldn't see evidence of that. So I think it's possible that maybe that gets triggered by some cell signaling event. Alternatively, it could be that really its function is to make really efficient degradation by reconfiguring the chains.
Thank you so much.
So Mandar, I have a question.
Yeah, please go ahead.
So very nice talk, Professor Walters. So I have a technical question. When you use NOE data, do you use it for yes and no of interaction, or do you actually calibrate NOE intensities for distances?
We do. We calibrate the NOE intensities for the distances. Yeah. So we for the intermolecular NOEs, we try, so the most important, so we certainly go through and give specific distances and calibrate it with the 1/r⁶ relationship. But we tend to keep in the back of our mind that if we make it too tight, that's really bad. So if anything, we kind of go on the side of making them a little weaker. So we like in some cases, because these complexes are so dynamic and because we're taking so many transients, we sometimes have to go up to taking 16 transients per increment. Then, you know, we kind of have the feeling that we might be seeing distances out to about 6 angstroms. So we do turn it into distances, but we definitely be very careful not to over confine any NOE, and we kind of always follow the rule that no one NOE should be so important.
Okay, thank you.
Kelly, I have one very quick question though. I mean, I don't see any others asking, so I can go ahead, probably take the liberty of being a chair. Yeah, so I just wanted to check with you about this RPN13 resonances. When it was bound to this diubiquitin in the extended form, did you see the signals being more broader compared to in the closed form, or did you not see any effect on the RPN13 resonances of the state of this open or extended form?
Well, I have to say the closed form, the signal is very strong, and I think because of population. So the signal, like for the free ubiquitin, you mean, right? So in the free ubiquitin, that closed signal is a lot stronger. And again, I think it's population dependent. I think it's because probably, I mean, we could have calculated it, but probably it's on the order of like, I don't know, I'd be guessing, but let's say maybe 75, 80% of it is actually in that closed state, because that other extended state, if you remember, the signal was fairly weak. And I think that's more of a reflection of probably population, that the dominant population probably is closed. That closed state is forced to kind of raise the bar on making ubiquitin less sticky. So you have to have a stronger affinity to kind of bind it. That's kind of what people have speculated.
Okay, thank you so much. Since there are no more questions, I thank you on behalf of NMR Society and my fellow NMR colleagues nationally and internationally. And now I invite the next speaker of this stellar session, that's Professor Mark Baldas. Professor Mark Baldas is currently the head and section NMR of the Bijvoet Center for Biomolecular and Research Center at Utrecht University in the Netherlands. He is a leading authority. Just one second. He's a leading authority in biomolecular solid-state NMR, famous for the membrane protein structure in native settings and cellular solid-state NMR. Since 2008, he's a full professor of structural biology at Bijvoet Center for Biomolecular Research at Utrecht, the Netherlands, as I mentioned earlier. He was a group leader before this at the solid-state NMR group at the Max Planck Institute for Biophysical Chemistry at Göttingen, Germany. Before that, he worked briefly at Leiden Institute of Chemistry, and he was a postdoc at MIT Howard Center with Professor Bob Griffin. And he did PhD at ETH Zurich with another two big NMR names, Professor Richard Ernst and Professor Bittmeyer. Some of the notable awards that he has received is this, the very famous 2014 Gunther-Lorchian Prize that was awarded by ENC to him. And another prestigious award that he received was in 2006 by the ICMR-BS Society, which was Founders Medal of the International Council on Magnetic Resonance in Biological Systems. Today he's going to enlighten us about sensitivity-enhanced solid-state NMR on dynamic protein assemblies. And with this, I invite Professor Baldas to give his exciting talk.
Yeah, thank you very much for this very nice introduction, and thank you for having me. I hope that you can see my slides. Is that correct?
Yes.
Yes, we can see the slides.
Okay. Yeah, again, it's a pleasure and an honor to be part of this, and I really enjoyed the talk of Kylie. So I hope I can, live up to this. Yeah, I started by sharing with you a picture of our city, because these days we can't travel much. So I thought I'd first show you something of the surrounding of our city. And some of you probably might have heard me last week at the DNP seminar. So I wanted today to talk about some more biologically oriented systems. You already heard the membrane proteins, and how sensitivity enhancements that we see these days in solid-state NMR, but also in some cases in solution NMR, is actually really allowing us to go way beyond what we have been dreaming of when I was, a PhD student or postdoc. And that has to do with the fact that we have, of course, now seen in the last 10, 20 years, really, especially in the solid-state NMR, a huge improvement in terms of sensitivity by, first of all, going to larger magnets, with, system, for example, here at our place. So this is actually a national facility that we have in the Netherlands. So these are the green arrows that you see here, actually the major leading labs in NMR and micro MRI in the Netherlands. We, about 10 years ago, we formed a national facility that allows us to coordinate our efforts, and also, for example, allowed us to get funding for a 1.2 gigahertz system that will actually be coming next year to Holland and to Utrecht. And so this sensitivity, coupled with the, the great work that many people like Bob Griffin have been doing to the solid-state NMR field, but also solution NMR of dynamic nuclear polarization, really made us think differently about the way we do biomolecular or material science applications. And of course, I'm not talking only about my lab, but many labs in the world that are profiting from these kind of advancements. Now, that was also one of the reasons, as you heard, when I moved about 12 years ago to Utrecht, that we realized, well, if we have these kind of possibilities, maybe we can actually, go into, systems, biological systems that are not amenable to classical NMR, certainly solid-state NMR techniques. So what I want to talk about today is 3 areas where I give you a little bit an idea of what we are currently trying to do with this kind of technology. I'll talk about our work on membrane proteins on the human cell surface. Then I will give you a little bit in an introduction and then some kind of report on what we do on human microtubules, which are a little bit different from what you would usually use when you use, for example, other kind of preparations. And then I want to show you an example, a beautiful my opinion, beautiful example where we combine Solus and solution NMR to look at a hot topic in NMR these days, where NMR shows the whole power of what it can do, which is liquid-liquid phase separation. Now, as I mentioned, when I came to Utrecht, we really thought, well, let's capitalize on these kind of sensitivity improvements that we see these days in NMR. And the reason for that was when we wanted to study membrane proteins, and as you heard, my group has been working on membrane proteins for a long time. We thought about the idea, okay, we can do the classical kind of purification strategy when you take bacteria and purify, detergent, solubilize, and then reconstitute. But why not live with what nature gives us, in the sense of that if you look at the bacterial cell envelope of an E. coli cell, you actually have proteins, but you also have endogenous proteins like lipoproteins, peptidoglycans, sugars, RNA. And so about 10 years ago, we started to think about ways to actually make these kind of molecules directly visible in bacteria. And so we started with a small enzyme called PARG-L that we wanted to study originally. And you can see here spectra where we compare cellular preparations with protoliposomes. We also studied a little bit, endogenous proteins that are actually expressed to high levels in bacteria, which is this LPP lipoprotein, which is attached here to the inner membrane of the outer membrane layer. But as I mentioned, you can also then, when you start to label bacteria and you analyze them, you can also see other strong components, endogenous components like peptidoglycans, LPS, and RNA that gives actually quite nicely resolved spectra. And over the years, we really further investigated how we can make maximum use of this. And one area, as I mentioned, is membrane proteins in our lab. And so, for example, here you see an example that we published 2 years ago. Lindsay Baker, a postdoc in my lab, she is actually a cryotomography person, and she's actually now doing that again in Oxford as a group leader. And so what she did was, when she was in my lab, she actually expressed a protein called YITC, which is a membrane insertion machine in the inner membrane of bacteria. So it sits in the inner membrane here in the white line of this bacterial cryotomography image. And what she could show is, to our great satisfaction, is that when you compare the resolution of a proton detect experiment of this cell envelope preparation of YITC, which is the green spectrum, it's actually quite similar to what you see when you actually purify, reconstitute the whole protein. And of course, the amount of protein that is needed for such kind of experiments is much, much lower when you directly use a cell envelope. And it was really nice to see that the same kind of preparations we use here for the NMR are giving also very nice cryotomography images, which gives us, and we are currently working on this, we're thinking about exploring this further, the possibility to actually combine the atomic information from the NMR with the tomography that is becoming, of course, more and more powerful. Now, another example in this kind of area relates to another insertion machine that we work on in the lab, which is a so-called BUM complex. I don't have time today to talk about this, but what you see here is actually one component of this complex. It's basically 5 protein machine of 200 kilodalton. And so what we did here, we actually expressed BUM-C, one of the components of this machine, directly to cell envelopes of bacteria. And so you see a spectra that we can obtain for these kind of preparations. And Shenji Zhang, when he was a postdoc in my lab, actually did beautiful work to actually assign, domains of these kind of BUM-C components. And that shows you that, we're getting close to resolution that we would see in vitro preps, but then, with preparations that are basically coming directly from the bacterial cell. Now, Sid Narasimhan, a really talented PhD and now postdoc in my lab, PhD student in my lab originally, went further on this. So I talked so far about membrane proteins, but Sid also developed our program of making, proteins in bacterial cell envelopes for membrane proteins to also study soluble proteins. And so what is important then, of course, is if you want to study a protein in the bacterial cell, in E. coli, for example, you need to make sure that the labeling only targets the protein of interest. And so over the years, we adapted methods that have been originally developed for solution NMR to induce expression, use antibiotics to basically target the labeling to the protein of interest, and then finally use DNP in this case for whole cells to study these kind of systems. And so that involves, transformation, targeted labeling, but it also involves, of course, really detailed protocol how to go from a, in this case, an NMRS solid-state NMR rotor, where you make your sample, you load it into a rotor, you mark your rotor, and then rapidly basically freeze and transfer this sample in the presence of DNP agents, radicals like amopole or others, to the probe of interest, in this case, a DNP probe in our lab. So this has really been a field of research in my group for the last 5, 6, 7 years, and we think we have now really a more or less established way how we can do that. Now, what Sid did as a post-PhD student in my lab was even more challenging, which is to say, can we actually study also proteins inside human cells? Now, we know the wonderful, fantastic work that has been done in cell solution NMR for the last more than 20 years now. And we also know that, of course, there are some limitations to that in terms of the fusion and the tumbling rate of the protein inside the human cell. So we thought maybe it would be interesting to have also a technique that allows us to study proteins in human cells by solid-state NMR. And of course, we can basically adapt the protocols like protein delivery by electroporation, for example. Veselenko has, for large degree pioneered this in cell solution NMR. So that is still available also for solids, and we did that. And once you have done this and you wait a little bit, then you can also then stimulate or treat the cells and analyze them by any kind of technology you want. But what was really crucial for the DNP in this case was to make sure that the radicals that we need to enhance the signal of these human cell preps actually rapidly diffuse into the cell without being reduced by the cellular environment. And so it turned out that we did a lot of microscopy experiment, and we used our friend ubiquitin, and I'm really glad that Kali talked about the system because it's a wonderful, I think, application of NMR and a very important application in structural biology, of course. And so what you see here is Sid's work where he actually made a fluorescently labeled version of ubiquitin, and he checked, in this case, I think HeLa cells it was, or HEK cells, I don't remember, I think it was HEK cells, the electroporation, after the electroporation of these cells. So what you see here in red are nicely ubiquitin populations, which are mostly seen in the nucleus. That's also what you would expect in the ground state, because ubiquitin, of course, likes to bind to chromatin and histones. And then we actually added a proteasome inhibitor that, of course, Kali knows very well, epoxomycin. And then indeed what we see here is, chain formation also in the cytoplasm of these cells, which clearly indicates that the ubiquitin that we made for NMR purposes is also fully active and functional in these kind of cell preps. Now, as already shown more than 10 years ago in cell solution NMR, is when you actually electroporate ubiquitin, wild-type ubiquitin into human cells, you will only see signals from the C-terminus, which is the most dynamic part of the protein. So that's exactly what we also saw in our NMR solutions NMR spectra. So you basically have fairly sharp signals from the C-terminus, but the folded part of ubiquitin is more or less elusive. And so there comes then the possibility to do solid-state NMR with this preparation. And there we could actually really, by 3D experiments, really trace a lot of the assignments of ubiquitin that we know from in vitro cases. So last year we had basically this kind of number of residues that Sid could pull out from the data, which seems to indicate that the majority of that protein is folded as you would expect, of course, from these kind of functional assays. And so that indicates to you that in principle, we should be able to study maybe also proteasome inhibition, or at least some aspects of that by these kind of techniques in the future. And of course, we are currently pursuing this. But originally, when we started to work on human cells, we actually were more interested in membrane proteins, so proteins that are on the periphery of the plasma membrane of human cells, because there, of course, we don't have this kind of challenge with getting the radical close to the protein of interest because they are staying on the outside. So this is work that started about 4 years ago when we looked at so-called A3431 cells, which are expressing high levels of the EGFR receptor. EGFR is basically a receptor that is activated by EGF. It's a small growth factor peptide or protein. And what we could do a couple of years ago is we could actually use solid-state NMR, sequential labeling, DNP, to actually investigate what happens when EGF, this is this yellow molecule that you see here, actually binds to EGFR in these plasma membranes. And so according to our data, I don't have time to go through this in detail, is that the receptor is actually quite mobile on the surface. That what we concluded on the basis of double quantum buildup experiments that we did. And then sure enough, when EGF binds, it actually, induces some kind of, structural change, which according to this field, most likely comes from the fact not only that EGF binds here, and that is known from crystallography, but also that these dimers or multimers are formed. And what we saw was that these dimers actually are more stable, more rigid than what we would see before EGF binding. So this is one of the aspects that we used, this kind of, label-based human cell preparation, so-called vesicles in this case. But what's even more intriguing maybe is that these receptors can be modulated in terms of their function by antibodies or nanobodies. So what you see here is, for example, the topology of this EGFR receptor and the dynamics that we have been observing in plasma membranes. So this is domain 1, 2, 3, 4 of this receptor that is on the outside of the plasma membrane. And these are actually the binding epitopes of a family of nanobodies, which are known to bind similar to EGF to this receptor. This is based on crystallography in vitro work. And what we find is actually, and this is recent work we've done in the lab, is that these nanobodies have different influences on the function and the internalization of the receptor. Because normally in wild-type EGF-bound cases, after dimer formation, there's an internalization taking place. The receptor gets basically internalized into the cells. And that's also, for example, what happens here if you have EGB4. This is one of these nanobodies that you see here. But then in other cases, this kind of transfer internalization process is stopped. And the receptor remains at the plasma membrane that you can see here. Now, what we have done now in the last 2 or 3 years now is now to think about how we can actually study this process by NMR and microscopy. And here you see a little progress report on this in terms of nanobody binding. So what we did here was we actually went to so-called 3D cell culture, spheroids. And if you want to know more about the preparation of these, I suggest to look at this paper here from Rainier Dammann, a PhD student in my group, which is now also graduated in the meantime. And what we then can see is how these nanobodies, in this case, penetrate the 3D cultures of these cancer cells. And this is the crystal structure in vitro that proposes how this nanobody binds to this domain 3 of EGFR. Now you can see this significant hole here, so I think there's still room for improvement on this kind of binding mode. And so what we did here now for using our 3D cultures is to actually isotope label in bacteria this kind of nanobody, and subsequently use DNP to actually make this nanobody visible under DNP conditions. So what you see is now here a spectrum of these preparations, and we did a number of controls to make sure that it's really bound by a nanobody. So we've washed the cells similar to what we do here. We use affinity assays and so forth. And sure enough, we can now see signal that would be consistent with the visibility of this nanobody. And so in the future, we want to investigate whether we could really understand the binding epitope of these nanobodies in these kind of cell cultures. So that brings me to the second part of my talk, which is again to do with human preps, cell preps. In this case, has to do with microtubules. And you probably know, of course, that microtubules are these kind of wonderful train tracks in the cell involved in organization and motility and so forth. And it has been, an interest for many, many years also for NMR communities. Now, in the case of solid-state NMR, I want to mention, for example, the work of Jana Polanova. She did really nice work on CREB-GLI, which is a microtubule plus end binding protein, and she investigated the structure of this protein. Now, in my lab, we also have been, interested in microtubules for a while now. So about 10 years ago, Ashutosh Kumar, who I'm still in contact with, and I'm happy, to be invited to this meeting also, he was studying when he was a postdoc in my group, studying small molecules like epithelon B as it binds to microtubules. So there was already quite, kind of interest for microtubules in my group for a while. But what we recently thought about is actually go more deeper into the interaction of so-called microtubule associated proteins or MAPs and their function. And so together with Anna Akmanova, she's a cell biology expert here at our university, we asked the question is whether we can, get more detailed insight by NMR and other technologies, how, for example, MAPs can discriminate between the minus and the plus ends of microtubules, but also how they interact with, for example, the termini and the tails of these proteins. And its first example where we did this is this so-called CAMSAP2 protein, which, as you can see here, binds to the minus end of microtubules. And that's also what you see here. So when you make movies, you see that in green that the CAMSAP stays on one of the sides of these microtubules. And it has this kind of domain architecture, so it has folded domains, but also IDP regions. And the most important one is this so-called CKK region. And so the question was how CKK binds. And then, of course, what you can do is you can use both solution NMR to look at the structure of CKK in vitro. And then secondly, we use solid-state NMR and AM microscopy, electron microscopy, to look at the decoration of this CKK domain once it's bound to microtubules. And the nice thing was that we could actually then use the assignments of the free CKK to directly study in the solid-state spectra the changes that we see in the chemical shifts. So what you see here are carbon-carbon correlation experiments in solid-state NMR, and the blue assignments that we had obtained from CKK in solution actually match what we see in the microtubule bound case. And as you can see from the red crosses here, there are some peaks which are missing or shifting. And so we identified these kind of shifts as, chemical shift or intensity changes. And if you plot this then on the cryo-EM structure that we had together with a lab of at UCL in London, you see that the EM densities, the NMR data really nicely fit together to enhance the resolution of this, density of CKK binding to the microtubules. So the red residues are exactly the ones that also are affected when compared to the free solution CKK data that we had. Now, the resolution of these complexes is not that high. And that has to do with the fact that CKK, as I mentioned to you, is actually a protein that selects for the end terminal, the minus end terminus. And so what happens actually, that if you, for example, do a mutation of these CKK domains, for example, by a single point mutation here at this position 1492, you see actually, again, beautiful solutions in the mass spectra of this protein. That's what you see here in blue. But now the resolution of the solid state spectra of the CKK mutant bound to microtubules is also extremely improving. Excuse me. So here you see now actually a proton detected solid state NMR experiment after binding, and the resolution is, as you probably agree, is almost comparable or even maybe better than in the solution state. And if you then plot these chemical shift changes or perturbations between these two states, they're actually quite small. It's about half a ppm at max if you look closely at the chemical shift variations here. And so for the moment, we were, of course, very disappointed at that. But thinking about the fact that, the binding of this protein is very tight, affinity is high. And importantly, what we also saw in CPMG, and that's work from Chenji Zhang when he was a postdoc in the lab, that this protein is actually extremely rigid on the NMR time scales, would indicate that this is actually designed to be fairly rigid binding, strong binding. And so that this binding actually leads to a contraction of the microtubule diameter, which was then picked up by the EM. So you see here in this kind of study that NMR and EM really nicely complement each other to understand how these proteins might recognize these specific regions of the microtubule surface. Now, I mentioned already at the beginning in this picture that microtubules also have tails, dynamic tails, which are all invisible to cryo-EM microscopy. And that's not only important in terms of the function of the microtubules, but it's also important for map binding. So these tails of microtubules actually are rich in post-translational modifications, and they are, as I mentioned, also very much influencing how these microtubule binding proteins actually interact with microtubules. And so with our experience of making human cell preparations for solid-state NMR, we got a little bit ambitious, and we also wanted to then make labeled microtubules from human cells. And so there we got lucky that we were working together with Carsten Janke in Paris, and he established actually a year ago a protocol how to produce microtubules from HeLa cells, S3 cells in suspension. And we adapted that protocol, which consists basically of lysis, cold spin, polymerization, depolymerization, and so forth, to produce C13 and 15 labeled microtubules. And then we made the microtubules, we checked them by electron microscopy before magic angle spinning and afterwards. And we were pleased to see that they were fully intact. And then we could start to do actually NMR experiments on these human microtubules. So on the left, you see actually now experiments which are based on so-called dipole experiments, so carbon-carbon correlation experiments using dipolar couplings. And you see, an alipharic region that is very much reminiscent of a strong protein, in this case, the microtubulin A and B. We could also do phosphorus NMR, and we could pick up signals from ATP, GDP, and GTP, which are actually known to be involved in stabilizing these microtubules. But then on the other hand, we could also do J-based experiments. So where we remove dipolar transfer blocks by J-based experiments, and they are particularly powerful to study mobile protein regions. And so this is what you get then, basically. So Shenji did these experiments, so C13 HSQC type experiments that we all know from solution NMR, but now under solid-state MRS high-speed conditions. And what you see here is this is actually the N15 HSQC that you get, which is, of course, a very simple experiment, but it very nicely matches with what you expect from the tails, because the tails are, as you can see here, the C-terminus and the C-terminus from alpha and beta tubulin are very rich in glutamate, some glycines. And the analysis of the data set really indicate to us that, yes, indeed, what we see here are actually the tails, which again indicates to you that these tails are extremely dynamic, and that's why they're not visible in any kind of microscopy data sets. So we actually managed to get from 3D experiment also some tentative assignments. For example, we could discriminate between EGE and FGE glycines. We could actually identify, also with help from previous solution NMR work in vitro, tyrosine 441, for example, and alanine 444 from peptides that were analyzed. So that was really quite assuring that what we see here is actually these kind of tails. But coming back now to the question, how these tails actually interact with the MAP proteins, what we then did was we basically repeated these experiments in the presence of CKK in this case, which I just mentioned to you. And here are the data. So what you see now is here that the spectrum that I just described is actually significantly changed. So this glycine correlations that we see here have moved or disappeared largely, whereas the EGE motif seems to be more or less intact, but we see also here additional peaks. What's also interesting is, for example, the arginine side chain signal is completely gone. And some other peaks are also gone, as you can see here. Now we have the cryo-EM data, so we can actually try to understand what's going on here. So what you see is actually now the EM data of CKK bound to the microtubules. And I highlighted here all the arginines, which are actually close to this binding epitope of CKK. And so what we think is going on, that once CKK bind, actually the side chains become more rigid because they probably get stabilized by interactions with CKK of the microtubules. Now what's also important, and I hope this is a little bit understandable here, is that because we cannot see any tails from either microtubules or CKK in the EM data, we in the moment have only approximate idea where actually the tails would be. For example, the C-terminus of beta tubulin would somewhere be this kind of dashed line that you see here in black. Alpha tubulin would be a dashed line starting from somewhere here. And then in the middle, CKK itself also has termini, as we can, that we know from other data. So what that would potentially mean is that actually that these tails actually interact with each other and lead to a modulation of this kind of signal pattern that we see here. Now, I fully agree with you, say this is a kind of a bold statement, but it comes from the idea that we then also took another MAP protein, namely MAP7, which is another one of this family of proteins that binds to microtubules and is involved in important interactions, for example, with tau also, in competition with tau. And here's the same approach again. So now we add MAP7 to these kind of human microtubules. And as you can see, now the spectrum stays more or less the same. The only difference we see is that the intensities vary significantly. For example, here we see a strong decrease in the alpha tubulin tails and also a strong decrease, except for the last one, for beta tubulin. And so if you reconcile that with what I just showed you for CKK, our interpretation is the following. Now for CKK, when it binds to human microtubules, we have an interaction on a pretty fast time scale between both tails, namely CKK tails and microtubule tails, that lead to this kind of modulation that we see. And in the case of MAP7, we actually have some kind of an on-off binding event where a significant time of our measurements, actually the tail is bound to MAP7 and then it released again to have this kind of an off tail. But of course, we have to do more experiments to classify and also to quantify these timescales. And that's not only relates, for example, to MAP7 that you see in this little cartoon, so bound here to the microtubule surface, but it actually applies to many other proteins that bind to microtubules. For example, tau is a classic example. Then they also not only, probably dynamic, but they also exhibit post-translational modifications, IDP regions, And last but not least, there's growing evidence that these kind of microtubule MAP systems, protein-protein networks, also give rise to liquid-liquid phase separation. And that brings me to the last part of my talk that I want to show you, namely the question, how can NMR, especially solid-state NMR, contribute to this kind of systems? And we started this project actually more than 10 years ago in context of the nuclear pore complex. And you may know that the nuclear pore complex, there's still debate what actually happens in the inside of this nuclear pore complex. And so a long time ago already, there was a big discussion in the literature whether it's going to be, some kind of an ordered selective phase of saturated hydrogel of proteins inside the nuclear pore complex, or it would be some kind of an oily spaghetti that you see here in this paper. Now, Dirk Görlich, a former colleague of mine from the Max Planck Institute, he works on NSP1, which is one of the main components of these nuclear pore complexes. And as you can see here in vitro, that these proteins actually form hydrogels. So they are beautifully, forming these kind of three-dimensional structures. And so we asked the question already some time ago, whether NMR, especially solid-state NMR, could be contributing to understanding what these hydrogels actually look like. And maybe you've seen a couple of weeks ago a new tomography study of a nuclear pore complex. And then I just took a little picture here where you see actually NSP1 here that is part of this rim of these structures that have been seen in tomography. And then again, the question is again, what happens here in this kind of hole in the middle? And NSP1 is supposed to be involved in the recognition and transport of proteins across this. So in our NMR studies, we actually used this protein made in vitro and then used, again, a very simple idea, namely dipolar and J-based experiments to actually find out what happens to this protein in this gel. And the spectra that we obtained, and that's already quite some time ago, is rich in different amino acids. So you actually see this different distribution of amino acids in the dipolar-based experiments, spin diffusion type experiments here, versus Subond Topsy type of experiments. And it turns out that most of the amino acids that show up here in this region are actually threonine, asparagine, and serine, which you see here. On the other hand, though, the mobile segments are dominated by more charged amino acids. And so if you look at actually the amino acid distribution, then it's actually very nice to see that these FG NSP1 proteins are very rich in these blue segments, so amino acids that are mostly found in the rigid spectrum that we found in NMR, whereas the charged aminos are actually part of the more C-terminal region of the protein that are not critical for hydrogel formation. So if you only take these charged amino acids, you will not form a hydrogel. And so what we could then also do in solid-state NMR is actually get an idea what kind of structures actually keep this thing together by doing mixed labels, protein samples. And what we found is that there's evidence for transient amyloid interactions between these proteins in these gels. Now, the reason why I show you this is, of course, we were really charmed by these kind of systems. And I was fortunate enough a couple of years ago to run into Remco Sprangers, which is a solutions NMR expert, as many of you know. And Remco's group has done really fantastic work on a so-called mRNA decapping machinery. And he has been working on this for a long time now, 7-8 years at least. And what he studies is this so-called DCP1, DCP2 complex, which are two proteins that interact with a network of mRNA decapping proteins. And I'm not going to go into details. Really, Remco is the expert on this, so this is all his work. But what is important is that he could show already in 2014 is that you can take DCP2 and EDC3, which is one of these interactors in this kind of network here, and you can actually form liquid lace fractionation. So you can see a vitro reconstitution of cellular phase transition process that involves, in this case, EDC3 and DCP2. And so, intrigued by and motivated by our work on NSP1P, I got in contact with Remco, and we decided to actually look at his system also by solid-state NMR. And what's really interesting and what Remco had in the meantime shown is that actually the EDC3 protein is sufficient to study liquid-liquid phase separation in this case. So what you see is actually microscopy images of this protein. I'll show in a moment what it is. And for solution NMR, you don't see any kind of phase separation. And then there is this, phase separated state. And then there's another state, which Remco calls the matured state, or what we call the matured state. And the only way to get to these kind of different states is by, for example, changing, and that is indicated here, I hope you can see this, by, for example, adding RNA. So when you add RNA to the system, you actually populate LLPS, but also this matured state, or if you change salt concentration. And so you have now a beautiful system where you can actually do both solution on a solid state NMR to interrogate these kind of transition events. And as I mentioned, so we started with the EDC3 domain, and this domain is actually, in terms of domain organization, given for the full length by 3 domains. It has an LSM domain, folded domain, as I'll show you in a second. Then there is a larger region which is actually intrinsically unfolded. And then there's another so-called YGFN domain, which is about, 250 amino acids, which is presumably folded. And so we started this protein as well as the deletion where we took out the LSM domain by both solution and solid state NMR. And solution state NMR was already done to LSD, but the solid state NMR data I want to show you now that we obtained in this kind of maturated LLPS state. And here we go.
You're exceeding your time, so can you please wrap up?
I'm almost done. Thank you. Yeah. So what I show you is now these kind of gels, and you see conventional carbon-carbon experiments where you see actually, if you take off the LSM domain, that the signals are gone. So that means this LSM domain, the N-terminal one that I just mentioned to you, must be dynamic. And you see actually also proton-detection experiments, which are similar to what you see in solution. So the green spectrum is the solution state spectrum, and in black you see the matured state of this protein, now studied by solid state NMR. And I guess you agree that most of the signals are very much superimposing well, indicative that the protein structure of this domain is more or less still intact even after this kind of phase transition process. You can even lyophilize this domain and rehydrate it, and then you also get the same result. So clearly what apparently happens is that in these constructs where we go through the LLPS, the protein conformation is preserved in these kind of states according to solid state NMR. What you can also do is, and that we did then also, we actually added RNA to these systems. And so what you see here are now solid state scalar-based experiments, similar to what I showed you for the microtubules, or dipole experiments. And the only difference is that you add RNA in the green spectra. So what you see immediately is that actually the J-based experiments that usually report on mobile protein components actually reduce intensity. So you lose intensity when you add RNA. And on the other hand, the dipole experiments actually increase in sensitivity. That's what you see here. So clearly, what happens is RNA somehow reduce the dynamics in this kind of mature state. And thanks to Remco's group, who has solution assignments for this IDR region, we could even pick out some of these amino acids which are probably involved in this stabilization of the IDR domain. And I don't have time to tell you much more about this, but what that basically led us to is a little model, a preliminary model. We're not done with this. What actually happens to this protein. So what you see here is basically what you see in solution for the ED3 domain, basically a dimer probably formed, a really dynamic IDR region, and then this LSN domain that probably moves. And what we think is going on, that LLPS and also the matured state, that actually this network formation leads to interactions, for example, between RNA and the IDR region. That we have solid state NMR data for this, what I just showed you. We also have an idea how the IDR actually interacts with the last domain, the YGF domain. And at the same time, we still see highly dynamic regions here of this LSM domain, even in this matured state. So that means the dynamics in this case coming at least for different domains of the system. And so with that, I'm finishing. So I wanted to show you a little bit our efforts in using sensitivity enhanced NMR, especially DNP, when we're talking about intact bacterial or human cells, spheroids, 3D cultures, and the possibilities maybe in the future to study with drug binding in these kind of cases. Then I showed you the use of human microtubular preps to actually study tail-tail interactions, dynamic interactions between the surface of microtubules, which are extremely important, and proteins that actually exhibit a lot of motion, IDR regions like the maps that I mentioned to you. And at the end, I wanted to show you our ongoing work. We still, of course, want to go on with this, how one could potentially in the future use solid and solution NMR to look at these kind of dynamic assemblies by studying both more rigid and more short time scale interactions and fast time scale interactions in the solution state. Now the work that I showed you was done in the first part by Sid Narasimhan, really talented PhD and now postdoc in the group. Alessandra Lucchini Paioni was involved in the DNP. The work on on microtubules was done by Chun Lu and Shenji Jiang, now associate professor at Hefei in China. And the last part was, as I mentioned, is a beautiful collaboration with Remco Spranger's group, and the PhD student in charge of that has been Rainier Daman, who's now working at an antibody company here close to the campus and is now taking over Bo Agnes. And with that, I'd like to thank you for your attention.
Thank you, Mark, for this wonderful, inspiring 3 stories that you have told us. We have 2 questions here in the audience. I see Grace Royappa is the first one. I request him to unmute the mic and ask the question.
Thank you, Mark, for that wonderful talk. I have 2 questions. I was wondering, can you hear me?
Yes, I can hear you, yes.
Okay, yeah. So in cell NMR, how do you know that this protein is in the correct form, not bound to any other binding partners known or unknown? That is my first question. And my second question is about microtubules. I was just curious about the small molecule vincristine. Do you have any insights on where would it bind to the microtubule?
Okay, I'll start maybe with the last question. You mean the small molecules that I mentioned to you, or the maps?
The small molecule vincristine, which you did not mention, which is used for cancer treatment.
Oh yeah, the apotolon B here, you mean this one here. This apotolon B.
Maybe it's called by a different name.
Yeah. Yeah, so this is, as I mentioned, this is actually 10 years ago work that Ashutosh Kumar was involved in, and at that time we were not able to make microtubules labeled. So that was before the time where we had only access to the ligand, and then we started the ligand labeled, and then we modeled how that ligand would bind to microtubules. Now with the technology that we have now and being able to human cells, you're absolutely right. It would be fantastic to now check how these molecules actually bind to microtubules, but we have not done that yet. I hope that answers your first question. And then the second question was about the conformation of the protein, right, in cell?
Yes.
Oh, just a moment. Yeah, so that's also a very good question. Well, I can tell you the following. So like I said, in the human cells, our studies that we have done so far, we have some other proteins, but we know most about ubiquitin is actually ubiquitin is bound. So if you look at ubiquitin, and you look also, for example, by gels or by other kind of other readouts, ubiquitin that we see here is actually strongly bound to nucleosomes and chromatin. So actually what we are seeing here, that the NMR signals are not coming from free ubiquitin, but probably ubiquitin that is mostly attached to something else. So you're absolutely right, we cannot separate between the two at this point because this is the first experiment. But in the future, we want to, correlate that with other readouts, for example, mass spectroscopy or other aspects in terms of what kind of structures we actually see, subpopulations, for example, adding a second protein, binding interfaces, and so forth. But what you definitely will see is the entire population of the protein in the human cell. For example, when we do this epoxomycin treatment that leads to chain formation, we see huge change in the spectra because as Kelly told us, then you have all these kind of chain formation events that will impact the structure of ubiquitin. And to my knowledge, nobody knows really how these structures will look like. And so we're really excited to find out what happens to these kind of ubiquitin structures. And as you probably know, ubiquitin is a pretty plastic molecule, even though it has a fairly rich structure. It exhibits a lot of dynamics, as of course NMR has shown us for 10, 20 years now. So we're really interested to find out what are actually the structural changes and adaptations that these proteins undergo when we put them in cells.
So Mark,
yes.
Another question by my student, Jaideep Paul. Jaideep, you can unmute yourself and start. Yeah.
Sir, can you hear me?
Yes, I can hear you.
Yeah, a very nice talk. So I have a question regarding LLPS formation. So which kind of residues are involved in this LLPS formation? And through solid-state NMR, can we map those residues or can we identify those residues so that we can understand this process much better way?
Yeah. So also a very good question. And I also have to disappoint you because we are not finished with this. Yes, what I wanted to show you is that we have now some idea what's going on. And that has to do with the fact that, and I have to there relate you to Remco Sprangers actually did mutations in some of the regions of the protein. So we have also, and Marcus Weingart, a colleague of mine here in Utrecht, he did some MD simulations to understand what we see here in NMR data. So we have, for example, some idea how IDRs, for example, interact with the binding with this folded domain. But we have not yet a complete structural model what happens here on a residue-specific basis. That's also why we are right now doing experiments to describe this further. So you have to take this a little bit, you know, as a first demonstration that we think with this kind of technology, you could go to atomic resolution information on how these processes are actually taking place. That we would be able, like you, I think what your question was, could you, for each residue, actually identify how they interact? And that's basically, yeah, one of the stages that we want to go now. But I think the technology, the combination, in this case of solid-state NMR, like we showed here, and solution NMR, I think offers really a lot of promise that you would be able to really decipher these kind of transitions as much as, you know, a conventional protein in the future.
So Mark, do you have a next question from your former co-worker, Ashutosh Kumar? I invite Ashutosh.
Please, Ashutosh.
Hi, Mark. So fabulous talk. Actually, I wanted to ask you about this nanobody organoid interaction.
Yes.
So nanobody being a bigger molecule, what kind of conformational change are we expecting if we are looking at the nanobody's structural change?
Yeah, so the nanobodies are actually not that big. Maybe you know that's about 140 amino acids.
Right.
And they have these kind of, wonderful loop regions, which are actually engineered to have a little bit more promiscuity of binding to different, regions of different receptors, for example.
Right.
So like you see in this X-ray structure, in vitro structure, there's this loop which kind of sticks out and probably is stabilized by another crystal contact here at this point. So I don't think we see, changes in secondary structure, but the question is how this loop and the domains here around this interaction are actually folded. So that's something we want to investigate, and where I feel, there's a lot of opportunity to really understand what's going on, especially when you are looking into nanobodies that are supposedly, specific for a particular protein. And maybe you've seen the last week's issue from Nature where there were discussions about the fact that, these antibodies and nanobodies might not be as specific as some people think. So I think there's a lot for NMR when you look at such a structure to gain to understand how they're actually working in the biological context. But, I don't expect that the strand here from this side, for example, would be changed. So that is also clear. But there's enough flexibility, and there are several loops here which are involved in recognition, which could be involved in this kind of event. And there's also, for example, this is not shown here, but actually many of the nanobodies bind to different domains of receptors. So you could also there see all kind of different conformational changes, which to my knowledge have not been investigated. We're not able to investigate by any technology to this point, unless you do crystallography or something.
All right. So the structure that, the spectra that you see in the right looks pretty broad. So this is coming from nanobody, right?
Yeah. So this is because it's under DNP condition. It's the first experiment we did to show that we can in principle see it. Mm-hmm. So in the next stage, what we will do is, for example, go to 800 DNP. That was 400 DNP. Okay. Maybe we can even do proton detection. We haven't, investigated this. Should also be possible with 3D cultures. And, of course, you can also change the labeling, right? That you zoom in, do 3D spectroscopy to really understand what's going on. This is just a proof of principle that you can use, these kind of organoid, spheroid kind of preparations. And then they're fully viable. You see here a control antibody that doesn't bind. But then you have now for the first time, within reach a situation where you get atomic information from such kind of systems.
Excellent. Thank you.
Mark, next question is from our host, Jitendra Chog. I asked you to ask the question.
Thanks, Mandar. A very nice talk, Professor Walters. So I have a related question which Grace has asked initially. So when you're doing solid state NMR on cells directly, so do you care whether the cells are dead or alive in the rotor? Would the protein conformations be affected if they're dead or alive, or like how do you manage?
Yeah, so for example, here we do this, I have not shown this here, but we also have done experiments at high temperature in these 3D cultures. And to our knowledge, to understanding, these cells are still viable, they're not dead. Now in the DNP case, you have to think about more like a tomography time experiments, because the DNP experiments are done at low temperatures, 800 Kelvin or so. And so the cells are fully viable until the final stage when we actually prepare the samples for NMR. So what we do is, and if you want, you can take a look at that paper, we really follow the cells, over the time after protein electroporation. We can, like I said, we can control them, add ligands, can do DNA damage, any kind of functional readout that you would like to do. And only in the very last moment, basically 5 minutes before the experiments, 5 to 10 minutes, we add these DNP radicals, and then we rapidly shock freeze these cells to do DNP experiments.
I see.
And in that case, I think I would say, well, they are trapped in some kind of a state where definitely they were viable.
Right. Okay. Thanks.
So thank you, Mark, for taking all the questions. In fact, I have a couple of them, but then I would write to you specifically, although I can't resist asking one question on liquid-liquid phase separation that you talked about.
Go ahead, please.
The LLPS that you have formed look pretty heterogeneous.
Yeah.
Where you're hinting them to be formation of P-bodies inside the cell. I remember from Roy Parker's images that they're reasonably homogeneous.
Yeah.
So any idea why your LLPS are pretty heterogeneous, and is it because the length?
You mean in the case of Remco systems here?
Yeah.
So I can't. This is. I would have to ask Remco about this, but I mean they are reversible, so you can always go back. Basically to this state when you change.
That's okay. And that's essentially to bring
but
back.
I mean, you raised an important point, and this is also still on my to-do list. My hope is, of course, at some point to do this kind of liquid phase separation inside cells. With the technology that I showed you today, I think we're in a position maybe to do this at some point in the future. But then we will probably see what really happens in cells by using labeled, I don't know, EDC3 or something. I mean, EDC3 is not a human cell protein, but other proteins, of course, many of them would qualify for such experiments. So that's actually.
Or you could do in yeast or something. Probably that's much easier.
Yeah, this is in yeast. Yeah, so my lab doesn't have experience with yeast, so I would hesitate to do this here. But I mean, I see a lot of potential to do actually NMR and then inside cells and compare to what you see in vitro in these kind of preparations. So I think there's a great area for NMR again in the future as well.
So with this, let me thank once again Mark Bartels and Kelly Walters for these wonderful presentations, both of them, and very live discussion post their presentations. On behalf of NMR Society, I thank them. And I think as a co-host of today's meeting, I have to announce next session meetings, but I'm not able to share my slides somehow. So
you can now.
Wait, Mandar.
now
Now you can. You have made a co-host.
it still says I'm disabled.
No, you are there now. You can.
Okay.
Okay. So wait a minute. Okay. It's complicated. Maybe it will go now. Okay, yeah, here you go. So before we end today's talk, I would like to announce that next week we will have two exciting talks by Dr. Sheetal Jain from UC Santa Barbara and by Dr. Rina Rosenzweig, who works at Weizmann Institute in Rehovot, Israel. And both of them are giving different perspective. First talk is about ESR spectroscopy primarily, and the second one is essentially about how protein disaggregation happens and how one can learn from these molecular chaperone machines that Rina will talk about to us. And with this, I thank organizers for giving me this opportunity, and also thank you for an extended session here.
Thank you very much.
Okay.
Thank you.
With this, we come to the end of today's talks. So let me again take the opportunity on behalf of NMRS to thank both the speakers, Kylie and Mark. And of course, thanks Mandar for chairing this session. So we'll meet again next week for two more interesting talks. Thank you. We'll call it a day.
Thank you.
Thank you.
Bye.
Bye-bye.
Bye.
Bye.
Bye-bye. Thank you.
Bye-bye.